Everyone, welcome to Cycle 4. We may not get through all the slides. Whatever we miss, we will pick up next week, and anything that we miss will not be on the quiz for this week. So this cycle is on DNA mutations. What we'll talk about are the types of mutations, the causes, and the mechanism of repair. So how is this mutation repaired? So type, cause, repair. Type, cause, repair, that'll be the pattern. So what are the different sources of DNA damage? So the focus of this lecture is mutations on the DNA. And so there are sources of damage, and they're categorized into two main categories, exogenous versus endogenous. So exogenous sources are more intuitive to you. You might be more familiar. We hear about this on a day-to-day, about sunlight and chemicals and ionizing radiation. These are sources that are coming from your environment, so from outside to inside your cell. Endogenous sources may not be as intuitive because it's not discussed on the daily. Endogenous sources are sources that are within your cell, inside your cell. There are molecules being produced, mechanisms occurring that are leading to mutations in your DNA. So let's turn to exogenous sources first. UV light. UV light damages your DNA by forming thymine dimers between the thymine groups in one strand or the other, but it's in these two adjacent thymine bases. So when this happens, the DNA polymerase will stall during replication because it cannot add the adenine base based on the structure that it's seeing. So when it stalls, it'll just come off the DNA and then maybe take over somewhere else. So you now have this section that was not synthesized, right? It can also just skip it over and keep continuing. That's rare. There are other things that can happen as well, but for the most part, it affects synthesis, which ultimately leads to mutation. Chemicals are in your environment everywhere. You inhale them, you ingest them based on your exposure, based on what you're eating. If you go to a grocery store, look at the back of a package, you can see all the ingredients that are in there. Many of them I can't even pronounce, so I'm not sure what's happening. A lot of the chemicals are actually banned in many countries, so it just depends on what you're ingesting. That's going to increase or decrease the rate of your mutagenesis in your cells. Ionizing radiation is also something that can lead to a double-strand break. Ionizing radiation is quite toxic because a double-strand break in the DNA, the cell goes into sheer panic mode, crisis mode, and it will do everything possible to stick those two strands together because the most unstable form in your genome is that of a double-strand break. So this can have deleterious effects. Endogenous sources. DNA replication errors occur, and that leads to DNA mutations. So for example, when the DNA is being replicated, so polymerase is replicating, and there is a mismatch. So when there's a mismatch, the DNA can be repaired. So polymerase itself can repair it with the help of either enzymes, or it can do it independently. So if it does it independently, that is called proofreading. So it'll go backwards, it'll remove the mismatch, it'll put on the correct base and continue. If it cannot proofread and misses the proofreading, then repair enzymes will be sent via p53, and that will be fixed. And that can happen during S phase or during the G1S checkpoint. So you have a lot of mechanisms that are there for safety to prevent this from happening, but it's inevitable, right? You could be living a very healthy life, but it's your DNA polymerase that is causing these errors. So as time progresses, you're accumulating mutations by all of these factors together. Another very important, probably the most important endogenous factor is actually your mitochondria. Your mitochondria does aerobic respiration, and one of the key processes in cellular respiration is the electron transport chain. All throughout your cell, all the metabolic reactions that involve electron or transfer of electrons, the electrons are flowing or moving in pairs because that's safe. But in the electron transport chain, the electrons move singularly, and that's very dangerous. It's dangerous because that will produce what are known as free radicals, and free radicals will produce reactive oxygen species, or ROS. ROS damage your DNA, RNA, protein. ROS, believe it or not, can lead to a double-strand break in your DNA, which is the most deleterious thing that can happen to your DNA, is both strands are cut. So they're pretty toxic to the cell. You are accumulating ROSs on a daily basis through time. So as you age, you have more and more ROS. And there's a lot of research going on in ROS and epigenetics and how both of those play a very key role in aging. So yeah, the electron is flowing singularly. So what the final complex does is it takes 4 electrons, gives it to oxygen, and then oxygen grabs a proton and it makes water, right? So that's the byproduct, it's safe. But what happens if that electron then just slips out and goes and reacts with oxygen? If that happens, then you get the formation of a superoxide free radical. Okay, this is actually an ROS. It's an oxygen species that will now become really reactive. And when it's highly reactive, it starts to react with your DNA, protein, and RNA, or it can then lead to further reactions that will produce even more toxic ROSs. So this will be converted to hydrogen peroxide. Hydrogen peroxide is really not as toxic. It's still considered an ROS, but it's not a toxic ROS because, you know, it in itself can't do very much damage. However, the most common reaction that happens with H2O2 is it can produce a hydroxyl radical. That is the most potent radical, hydroxyl radical. So these are highly reactive. The reason they're damaging your DNA is because they want to react by getting rid of their unpaired electron. Why are they so reactive? Because they want to reach stability. It just comes from Gibbs' law, where molecules, all molecules, you know, in the world want to reach minimum free energy, maximum entropy, you know, most stable, maximum stability. Who doesn't want to be stable? Molecules want to be stable too. So stability is their goal. To be stable, they need to get rid of that unpaired electron. So they will react with anything that's around. There's DNA in your mitochondria, as you know, so that's a target. It will react with that, with proteins, RNA, whatever it can find, because those are the ones that are abundant in your cell, right? In your mitochondria, it has all of these molecules around. However, there are ways to get rid of these ROSs, in other words, convert them to a safe molecule. So there are reactions that take, for example, the hydrogen peroxide and ultimately convert it to water, or the hydroxyl free radical converted to water, or the O2 to water. So water is sort of a common end product that these free radicals get converted to. And who's doing this conversion? So in other words, who's protecting you from accumulation of ROS? These molecules are called antioxidants. Antioxidants. You have natural antioxidants in your body that you were born with, you've always had it, and even if you don't eat and ingest your antioxidants through your diet, you probably will be fine. I mean, you do have some. But of course, the more antioxidants you have in your diet, the better you'll be off with reducing the amount of ROS damage. So yeah, antioxidants then basically take that electron and then dissipate it, and now you are safe from that. So basically, this is the summary of what we'll talk about, and we'll just go into more detail. So what are the types of mutations? Substitution, deletion, insertion, and inversion. Substitution is also known as point mutation, which is the way I will refer to it because that's how it's referred to in journal articles. I rarely see it written as substitution. A point mutation is when you get one base pair change. So here, This is the normal, but we seldom ever call it normal. We call it wild type. Wild type is the most common sequence in the population. The word normal is not something we use in genetics, so wild type. If you compare this to wild type, you can see this is AT, and this is now GC. So this is, at one point, there has been a change. The change is in both bases, obviously, because of the basic natural structure of DNA. It's a helix. So if one base changes, the other one has to change too. So if A changes to G, then naturally T has to change to C in order for this helix to function properly with the proper complementary base pairing. However, this is a mutant because it was supposed to be an AT, it is now a GC. This could easily code for a different amino acid, or it could not, depending on what codon this will lead to. Deletion means that you literally have base pairs deleted when compared to the wild type. Insertion means that you have extra bases added that were not supposed to be there compared to wild type. That's an insertion. And inversion is when the sequence is flipped. Before we go into mutation and damage, we need to understand the difference between DNA damage and a point mutation. DNA damage is a single-stranded change, whereas mutation is a double-stranded change, and it needs to be complementary base paired. So here you have a DNA strand. So just put your thinking caps on, or imaginary caps, I should say, and let's try to separate these strands. This is now going to divide, right? So the cell is going to divide, so DNA synthesis is going to occur. Look at the bottom, that is now going to serve as template. So when polymerase comes and adds the bases, it adds a G instead of a T, so that's a mismatch, right? This is a mismatch. And so what's going to happen in the next replication round, when this separates, this right here will serve as template, and when polymerase adds and makes the synthesis of the new strand, it adds the C. Well, it sees a G, it doesn't have the sequence memorized. It's not going to stop and go, oh, wait a second, you were supposed to be an A, get out. No, it sees a G, it puts a C. That's the job of polymerase, just to complementary base pair with what the bases it sees. It cannot memorize the sequence of 3.2 billion base pairs. So you now have a mutation because the wild type was TA. This is GC. So look at this, double strand change, complementary base pairing. Anything else, guys, is DNA damage. So on a test, don't put mutation for something like this. This is not a mutation, this is damage. This is a mutation because the mutation goes undetected. It is now a mutant. It'll keep dividing and replicating and doing whatever it has to do to cause a disease, for example, and no one's recognizing it. P53 is like, yeah, go ahead, you look fine. I mean, it's GC go together. P53 doesn't have the sequence memorized. But P53 can notice this. Polymerase can notice that, right? So mismatches do get noticed, and they get targeted, and they get repaired. So this is called damage because the cell is now prone to mutation. It is vulnerable to mutation. In the next replication round, as you saw, it became a mutant. Mutant is very dangerous. A cell damage, to be honest, is not as dangerous because it just kind of sits there like that. It can just sit as a damaged cell, or it can be repaired. The repair process can happen during S, which often it does during S itself, and if that gets missed, it'll be repaired during the G1S checkpoint, and that'll be driven by P53. So let's look at the repair mechanism. You get a mismatch, and now you need to repair it. There are two ways to repair it: proofreading and mismatch repair. So proofreading is something done by polymerase itself independently. It doesn't need anyone else. Whereas mismatch repair is done by polymerase with the help of repair enzymes. So it's teamwork in mismatch repair. So here is the replication fork with the DNA polymerase polymerizing, and it adds the wrong base. That's the wrong match. So here's what happens. The first way to fix that is through proofreading. So as DNA is synthesizing, it encounters an A. It's supposed to put a T, but it accidentally puts a C. So that's a mismatch. This mismatch is sensed by polymerase itself. You can see the structural change. You can see the distortion in the backbone. Polymerase itself will recognize that mismatch, and then it'll start to move backwards to remove that base and then move forward again. So in order to move backwards and forwards, these enzymes have to have directionality, and it has to have domains that can move backwards and move forward. So here's a structure of DNA polymerase. It actually looks like a hand. That's why they called it the palm, the thumb, the fingers. And over here you can see this particular domain that it has. So enzymes, or proteins can have different domains, and the different domains have different functions. When it moves backwards, it does so by using its 3' to 5' exonuclease activity. Exo means that it's at the end, not in the middle of the DNA sequence, somewhere in the end. And nuclease is an enzymatic activity that removes nucleotides. It literally removes them. So it moves backwards. When you're working in a lab and you want to design an experiment or design a new enzyme, you need to understand the different domains that you want the enzyme to have for your experiment, and you need to understand that you want it to do certain things, like move forward, move backward. So understand that what type of enzyme you're going to order or what type you're going to make to have those different types of domains. So for this, you need an exonuclease activity. You need the 3 to 5 domain. It also has a 5 to 3 synthesis domain. In other words, it moves 5 to 3 to synthesize. It moves 3 to 5 to remove the nucleotides. It needs 3 to 5 exonuclease for proofreading and 5 to 3 for synthesis. There are also enzymes that are called endonucleases, and endonucleases are ones that can go right in the middle of a DNA and start removing bases, like boom, 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 right in the middle. Exonuclease can't do it in the middle of a DNA strand. It has to do it at the very end. So polymerase has exonuclease. So it moves backwards, removes the C, and then its synthesis domain kicks in and it starts to move forward. Okay, so it's like the brakes and the reverse and drive in your car. When it moves forward, then it adds the correct base, and now you have AT. That was what it was supposed to be. So it senses this. And proofreading is done immediately. So as polymerase is moving, it adds the wrong base, it reverts back right away. It's not going to keep synthesizing and go, oh, wait a second. We got to go back to that CNA that I missed a long time ago. It can't happen. It has exonuclease activity, not endonuclease. So it has to detect that right away and start pushing backwards right away. Now, what if proofreading gets missed? What if polymerase keeps going and that mismatch is still there, which often happens, right? And when that happens, then the mismatch repair mechanism kicks in, and this is driven by P53. So P53 will send out repair enzymes and say, okay, there's a mismatch, I can sense it, there's distortion in the backbone, we have other enzymes that are telling me that something's wrong here, and I need you to send enzymes that will now go and scan. They literally scan your genome, guys, and they look. So P53 will say, I don't sense a mutation, but I still want you to go scan for them, right, to double check. So it'll scan and go, T and G don't go together. So these enzymes will make cuts in the wrong strand, in the newly synthesized, and it will take it out. And now polymerase will come, recognize that 3' OH overhang, it'll synthesize, then ligase will come and seal that nick, and now you have the proper sequence. This is a great example of teamwork. A common question I get from students is how do the enzymes know which strand to cut? That's a great question. For purposes of this course, without overcomplicating this lecture, just understand that the DNA has markers that the cell recognizes. These molecules recognize. So prokaryotes have different markers, eukaryotes have different markers. They recognize which strand is new, which strand is old. But if you're interested, come and ask me during office hours. They cut the correct strand and then they synthesize. Can they correct the wrong strand or cut the wrong strand? Yes, it can happen, but don't worry about it for this course. Just know that it happens. Okay, so we learned how you can get a point mutation through mismatch, right? If a mismatch goes unchecked, that's damage. And then in the next replication round, you get the mutation, right? Double strand change, complementary base pairing. So now what happens if you get a double strand break? This is a type of, a mechanism of mutation. In other words, how are these mutations caused? So ionizing radiation, we mentioned, and ROSs, both can lead to a double strand break. When the DNA senses a double strand break, it goes into panic mode, and its first instinct is simply to put those strands together, just glue it back together, because the genome instability, it just can't handle it. And so what it will do is it'll just stick it back together. But here's the problem. It doesn't have the sequence memorized. It doesn't know that this was ATG in the wild type. It just goes, oh, I don't know what it was. Let's just stick it back together. Let's just pick up some random bases or get rid of some random bases. I don't know, but let's put this thing back together. And in doing so, it leads to a mutation. And these mutations can be deletion, it can be insertion, it can be inversion, just depending on what is picked up. So in your nucleus, you have bases, right? You have bases just hanging around. Even if the DNA is not being replicated, these bases are still being used for other reactions. So these are all these bases around. So it can, in this case, it's a deletion because it forgot the G, but it didn't forget, it doesn't know what the sequence is. So it puts AT, now you have a deletion. So this type of repair is called non-homologous end joining, NHEJ. It's very sloppy, and more than often it results in a mutation. Can this go back to wild type? It sure can. It can go back to wild type, but that would be just luck. It's sheer luck. So in a lab, when you're doing these experiments and you look at some cells, you're like, oh, these ones reverted back to wild type, that's awesome. But for the most part, most of them are mutants. It's called non-homologous end joining because it is not using homology-directed repair, HDR, which is something you will learn next term. For this term, it's just NHEJ. HDR is when it's using its homologous chromosome to copy over the sequence. In this case, it's not. This is just joining up together. It doesn't have template. If it adds more bases, you get an insertion mutation after NHEJ, and you can also get inversion. But keep in mind that you can also go back to wild type. So if you see a wild type sequence, I'm going to go, wait a second, but I thought NHEJ only leads to mutations. No, by sheer luck, it can happen. If you break a cookie in half and all the crumbs fall, it's almost impossible for you to pick up all those crumbs and put them back together, right? So it's, for cells, it's the same thing. It doesn't have it all memorized, but in the off chance, it can go back to wild type. So that was one mechanism of damage and repair that you saw. So damage through ionizing radiation, or ROS, double-strand break. That leads to insertion, deletion, or inversion. It could also go back to wild type. Okay, now let's look at the next type of mutation, which is indel, and the cause. So the mutation is very similar, right? Insertion, deletion, but what causes it? In the previous example, that was a double-strand break that caused the mutation. In this example, it's DNA polymerase itself causing the mutation. So it does it by slipping during replication. So it's very similar to you running and falling forward or falling backwards. It's just slipping. And so it will slip during replication. Here you have your normal cell or your wild type cell. The green is your template and the red is a newly synthesized strand. So this one you have to pay a little bit extra attention and follow this. I want you to understand exactly how it is that you end up with a deletion and an insertion. If polymerase slips backwards, in other words, if it's synthesizing and it moves backwards, then you get insertion mutation in the following way. It's synthesizing, imagine, and it moves backwards, so it slips backwards. When it slips backwards, the part that is synthesized already, it can't see it. So don't worry about these bases looking like like they're codons. They're just written this way so that you can follow the number of bases, okay? But this is DNA, this is not RNA. So it slips backwards, and this here forms a loop. It can't see this loop. And so it keeps making, it says, wait a second, I thought I did this third one already. I'm pretty sure I did it, but I don't see it, so I probably didn't do it, so it does it again. And now, look at this strand is longer than it's supposed to be. And so in the next replication round, this will become template. So now the template in this mutant cell is longer. This is the wild type cell, this is the mutant cell. Well, it doesn't know that it's mutant. It just says, well, the template, I have to follow the template. The template is what I have to copy. So it copies it, but while copying, it has now made a cell with an insertion mutation. With respect to forward slippage, you get a deletion mutation. So here you have, again, your template Forward means that it skips reading part of the template. In other words, it skips forward. It doesn't even see that it forgot to read this one. So when it skips it, these ones are not complementary base pairings, so their structure changes. They complementary base pair with each other. They form a loop. They're kind of lost. Where's my partner? Where's my pair? So it just kind of sits there in a loop. So it skipped it. When it skips it, look at this. In the next replication round, the synthesized one is shorter. So now this is the mutant, and this is the wild type. The wild type was supposed to be this long. That makes sense. So you have one wild type and one cell with a deletion mutation. This is not damage. These are actually mutations. So on an exam, I will want to know the outcome of one replication round or two replication rounds. So when you have one replication round, you get 2 cells, 2 replication round, 4 cells. If you want to memorize just slightly, just to get you started, students in the past memorize backwards leads to insertion, forward leads to deletion. They use bi as, like the word bi, not a word, but like the bilateral, biweekly, bi is backward leads to insertion, and the other one is FD, forward leads to deletion. But I'm not going to ask you a question on what does forward do, what does backwards do, obviously. I'm expecting you know that. I'm going to ask you the outcome. What is the outcome of this slippage after 2 rounds of replication? How many cells will have deletion? How many cells will have wild type? How many will have insertion? How many wild type? So that's the way I want you to think of this slide when you're preparing for the exam. The next concept we want to cover is how do you get point mutation via tautomeric shifts. So this is another way in which you get mutation. There's so many ways you get mutation. Isn't it scary? Like how many? It's like we're all doomed from the start, right? So you have bases that have their keto form and enol form, amino form, and imino form. So what that means is thymine and guanine, they have a dominant structure and a rare structure. So its dominant structure is known as its keto form, keto, and its rare structure is known as enol form. So it tautomerizes or it changes structure between enol and keto. It mainly stays in keto, but sometimes it'll tautomerize and become a tautomer, and it'll be in its enol form. Cytosine and adenine are dominantly in their amino form, but in their rare form, they can tautomerize to their imino form. Here's the issue. When they tautomerize, they have a different preferred partner. That's what makes this lead to a mutation. So thymine, in its enol form, prefers guanine. Cytosine, in its imino form, prefers adenine, as opposed to A here and G here, right? So once this match occurs, that in the next replication round, that leads to a mutation. So when synthesis is occurring and polymerase sees thymine in its enol form, it literally goes and it grabs guanine. It doesn't grab adenine, because you're like, but T goes with A. But T is tautomerized. So when T is tautomerized, it's going to grab the guanine because it goes, oh, you prefer guanine. I'm going to give you guanine. But it's not supposed to be like, no, don't give it guanine. Give it adenine. But it's saying, well, it's tautomerized. I don't know. I don't recognize it in any other form. It's not keto. So once you know this concept now, I'm going to go through an example of how I will ask a question on the exam. This is just a chart to show you, what the preferred partners are. Don't memorize it. On the exam, I will tell you what the preferred partner is for these different tautomeric forms. This you would have to know AT, CG. That's about it. So let's go through this example, which is a type of example you would have to do on a test. So I will give you a cell that is starting at some point. It could be in its wild type form, it could be in its damaged form, it'll be a type that it's sitting at. And then I will ask you what the outcome is after X rounds of replication. So either 1 round of replication or 2. I'm not going to ask you 3 rounds of replication. That's 8 cells. That's too much, okay? So that's 2 cells, 4 cells, 8 cells, 16 cells. So just know the first and second round. I may throw in a third round. I doubt it, though, but let's see. The bottom line is just focus on the first and second round. Here you have your wild type parental strand, GC. There's a spontaneous tautomerization event that occurs, and that's denoted as star. So whenever you see a star above a base, that is its tautomeric form. G and C are here together because, well, it's not replicating, so the T will not be there, right? So right now in your body, your bases are tautomerizing. So a G somewhere in your genome has gone to G star and is sitting there at G star. It's not doing anything, there's nothing wrong with it. And it'll go back to G. But if it's in G star and replication happens, you're going to have a problem then. So here you have G star C. Do your first replication round. So when I ask you what the outcome is after first replication round, you have to do the round. So to do it, I would take this template, put it up here, take that template, put it down here, and these two will be your synthesized strand. So now pretend you're polymerase and add the bases, right? So this is kind of obvious. These are all complementary based. So this strand will produce a wild type. This strand, however, will not produce a wild type. It'll produce a damage. This is not a mutant, guys. This is not a double strand change. Sometimes you'll see a double strand change. In this case, it's a single strand change, so you'll say this is damage, but sometimes you'll see a double strand change. But remember we talked about both criteria have to be fulfilled. Double strand change, complementary base pairing. This is not complementary base pairing. So what you have to do is the following. G star, I will tell you that it has to go with a T. So you're going to put a T there. Now copy this over for the next replication round. It's here. And you're going to do the next replication round. But before that, in the question, I might say that before the second round of replication, G in its enol form reverts back to its keto form. So G star reverts back to G. So you would have to erase this. Don't draw this whole thing again on the exam. It's a waste of time. Erase that star, and now I'm saying, okay, before you replicate the second round, I want you to know that it is no longer G star. It has tautomerized previously, but it is now back, it reverted back to its dominant form. So you're like, okay, got it, perfect. So I start off with this now. So now let's copy this over in the next slide and take each one of these DNA strands and replicate them, right? So then you're going to have 4 cells in the second round. So this one is going to look like this, and the bottom will look like this. Don't draw this out on an exam. 1 wild type is going to make 2 wild type. Yes, 1 wild type makes 2 wild type. So just write 2 wild type, right, next to your wild type sequence. DNA damage, take this top, put it here, this bottom here. If you do top and bottom, it's important. If you flip and flop, you're going to get the question wrong, and you'll see why. When I ask you if it's a transition versus transversion mutation, you'll get it wrong. Don't flip and flop. Keep it all consistent. This top strand here, this bottom strand here. Now, this is synthesis. So this top strand leads to wild type cell. This strand, however, finally you get your mutation. That's your mutant. Because this was T, it's not a tautomer, it's a regular T, it's a normal T, and then you put an A, this is the mutant. So the answer is, when you had that situation, that parent strand, GC, and you had tautomeric events occur, the ultimate result after 2 rounds of replication is a production of 3 wild type cells and 1 mutant. That's your answer. It's not damage, it's not 2 wild type and 2 mutants. This is the way, so you have to work it out. Now let's go back. What if in the question I said that this happens, sure, you do your first round, but I say that G star remains as G star. It doesn't revert back to its dominant form. It remains in its tautomeric form. How will that change then the progeny in the end? This will be damage, mutant, wild type, wild type. If you have to do the question right, and you guys know on the multiple choice, right? It's going to be 3 wild type, 1 mutant, 1 damage, 1 mutant, 2 wild type, etc. So make sure that you do it consistently and keep the strands correct for you to get the correct answer. All right, so now this is just a summary of what we just talked about in all those slides. If that was like, oh my God, there's too many slides, this is all done for you right here. On an exam, you don't need to draw all this out. Just know that 1 wild type is going to give you 2 wild type. 2 wild type will give you 4 wild type. So that's just something you should just know. All right. Speaking of these tautomers, there's also something else that can tautomerize, and those are mutagens. Some mutagens are tautomerically unstable base analogs. So this is what this means. This is just an example, uracil and thymine. Uracil, this is the structure. The only difference between uracil and thymine is this group here, methyl versus the hydrogen, and that distinguishes the uracil from the thymine. Uracil is in RNA, thymine is in DNA. However, look at this. This is 5-bromouracil. This is a mutagen. It causes mutagenesis, which means mutations in your DNA. And just because of this bromine group, you were thinking, well, can it tell that it's bromine? Well, no. In this case, it can't. It sees 5-BU as thymine, and that's dangerous. So it will actually take 5-BU instead of thymine and put it on there, depending on the concentration levels, right? If you have more 5-BU than thymine, it'll pick up 5-BU. So 5-BU is a mutagen. And this mutagen is usually used in the lab to induce mutagenesis. So if you want to do an experiment, we want to induce mutagens to study them. But you guys know that there are mutagens in our environment that's causing mutations of our DNA. And they're still being discovered, right? You hear in the news, this causes cancer. This is how is it causing it? Part of it is because it's labeled as a mutagen. So it will cause mutations in the DNA, or mutagenesis. So both T and 5-BU have dominant keto and rare enol forms. So both can tautomerize. So let's do an example. So now that you've learned all that, let's do the next example. You have your wild type. You have this round of replication. So you separate the strands. And if you have a higher concentration of 5-BU, polymerase takes 5-BU and puts it on in place of T in its keto form. Then what happens is you get tautomerization of the 5-BU, and it converts to its enol form. In its enol form, 5-BU prefers guanine, and that will be in the stem of the question. I will tell you that 5-BU in its enol form prefers to bind with guanine so that you will know what base to put there. This top strand will lead to wild type. This is the one that you're concerned about. So you're going to separate these again. Is this damage? Absolutely. Is this mutant? No, it's not a mutant. So now, here's where the mistake happens. Students will say, but you said that mutation is a double-strand change. So AT went to G5-BU. Is that a double-strand change? Yes, it is. But did it satisfy the second criteria? No. What's the second criteria? Complementary base pairing. So separate this now. Now, there you go. That's your mutant. GC, AT. Compare that to that. That is your mutation. This is a damaged cell, and then you're going to get your wild type produced as well. So you have transition, mutation, damage, and wild type. Why is this called transition mutation? This is why. I'm going to ask you in the exam what type of mutation it is. So you're like, okay, so I just learned how to define a mutation. I told her this is a mutation. Now she wants me to go one step forward and say what type of mutation it is. Absolutely, I do. To know that, you have to memorize just something very simple, not this whole entire thing. All you have to know is that adenine and guanine are purines, cytosine and thymine are pyrimidines. Just find a way to memorize it. If the bases change from one type of purine to another type of purine, or one type of pyrimidine to another type of pyrimidine, that is a transition mutation. But if the base change is from one type of purine to one type of pyrimidine, or vice versa, that is a transversion mutation. So transition and transversion. So if you go back to this, this was transition because I'm comparing A, which is a purine, to G, which is a purine. If you take the bottom strand, you'll get the same answer. T is a pyrimidine and C is a pyrimidine. So purine to purine or pyrimidine to pyrimidine, that is transition. If you keep your strands the way you're supposed to, top stays on top, bottom at the bottom, you will get the correct answer. But if you flip and flop, you'll get the wrong answer. But if this is flipped, then you will say it's transversion, right? So don't flip and flop it, just keep all the top strands consistent, the bottom strands consistent, and you'll get the right answer. So we can't leave this lecture without learning about transposable elements and how they can lead to changes in the genome. And do we call them mutations? Sure, we can call them mutations. We can say that transposable elements lead to mutations, but often transposable elements lead to variation, and so you just get a different, phenotype. But let's just say for purposes of this course, let's just stick that yes, TEs can lead to mutation. So what are transposable elements? They're also known as jumping genes. They were first discovered as jumping genes, and they're now called transposable elements. These are sections of the DNA or the genome that literally move. So they can cut and paste, or they can copy and paste. So they're actually jumping from one region of the DNA to another by cutting and pasting or copying and pasting. So Barbara McClintock, a geneticist in the 1930s and 40s, she discovered transposable elements. And in the 1930s and 40s, there were very few women in science, especially in genetics. It was a male-dominated field. And if you look at her biography, and you learn that she really struggled during her education, and when she came on faculty, she really struggled because she just wasn't respected as a female scientist. And it was male-dominated, and so she finally got really frustrated, and she just, she went off on her own. She just said, I will do research on my own, by myself. And so she moves, and then she starts studying, and she discovers transposable elements, and she wins a Nobel Prize. And this was the first solo Nobel Prize for this category. And she won this in a solo manner. And they've discovered since then that all organisms in the world have transposable elements or jumping genes. But she called them jumping genes. Can you imagine? You already don't have the respect of your colleagues, and then you say, I'm going to do research on my own, and you come back with a discovery and go, guess what, guys? I discovered something. They're called jumping genes. They're jumping around. They're like, okay, so she's crazier than we actually thought she was. So yeah, she won the Nobel Prize. And you know what she said? She said, one day I was just curious. I was looking at corn. And she was looking at corn, and she was saying, why are the kernels different colors? And she said, I know, is because there are portions of the genome jumping around, and they're interfering with the biosynthetic pathway of the pigment. So this is the TE, transposable element. When it jumps, that's called a transposition event. It undergoes transposition. So here's the gene to make the pigment purple, the purple color. Purple color itself is a pigment. It's not coded in the DNA. It's not a protein. It's not an RNA. It's a compound, right? But pigments are made through enzymes. Enzymes are coded in the DNA. So what she said was, look, the TEs are jumping and interfering with the expression of this enzyme that is involved in the biosynthetic pathway of the pigment. So when it interferes and doesn't make this enzyme, then you get a colorless corn or yellow corn. But when this TE excises itself out, then you go back to purple again because that gene is working and it can produce the pigment purple. So yellow corn here, purple corn here, yellow, purple. Imagine how much this TE is jumping around in that single kernel. Isn't that incredible? So yeah, TE is found in all of these species. So here, even in a single kernel, you can see the yellow part will have this genotype, and the purple will have this one. So TE is in this gene. When it's yellow, that means no pigment production, and when it's out of the gene, then you get pigment production. Do you remember in cycle 3, we talked about C-value, and we looked at the C-value of plants. They had such a high C-value. Well, you know why they have it? Because they're packing in transposable elements through evolutionary time. Transposable elements play a critical role in evolution of your genome. They can rearrange your genome, guys. Like, it can literally pick up an entire gene and move it to another chromosome. So rearrangement is happening, right? It can do that. So in plants, it's very common to see TEs. And that's why you see such a high C-value. Look at barley, 5,000 mega base pairs. Like, do you think barley needs that many base pairs compared to rice, 350? I don't think barley is that much more complicated than rice. Corn, 2,500. But what is it that's packing it in is transposable elements. Look at barley, maize, and rice. Same number of genes, just a cartoon picture showing 3 genes. Gene A, B, and C. A, B, and C, A, B, and C, all of them, but the genome size is different because of the number of transposable elements. And these TEs are very active in plants. They jump around. Guess what? We also have TEs. 50% of our human genome is made up of transposable elements. They're still there from evolutionary time. But here's the interesting thing. This is how our TEs work. Here you have your gene. The dark regions are exons. The light green are introns. Look at the location of our transposable elements. They're all in safe havens. They're all in safe places, i.e. the intronic regions, and they're not interfering with the regulation of the gene, i.e. the splicing, the alternative splicing, all that stuff is all intact. They insert themselves in ways that it is not interfering gene expression. That's how we evolved. Look at that. 50% of our human genomes are TEs, but look at the way they're located in our genome. It's so beautifully located that it doesn't interfere. But more importantly, the location is not as important as the function. They can be located somewhere. What if they're undergoing transposition, right? Because then you're interfering with your gene expression. Look at the TEs. The TEs are dead. They're not functioning. They're inactivated. So we have evolved to inactivate our TEs. They're in safe havens. They're in certain safe havens, but also they're dead. They're non-functional. And so we, as hosts, have silenced our TEs, so they're no longer active. They can't jump around. However, the scary part, they retain the potential to be activated. So our silencing mechanism is there, but if that mechanism starts to fail through mutations in our genome, then you start to see that release of that inactivation, right? And now the TEs are like, oh, maybe I can jump around. So the TE will jump. It jumps, and then it will interfere with gene expression. And next week, we'll talk about how transposition leads to certain diseases for us. We'll skip this slide, but we'll talk about this one. ALU is the most common transposable element in our body. We have 1 million copies of ALU, and ALU can jump. So ALU has been shown to jump around in your genome and lead to transposition, and it can cause cancer and other diseases, which we'll learn next class. And you will see that although through evolutionary time this has benefited us, right? We have a rearrangement of our genome to benefit evolution. It can also cause diseases because it still has the ability to undergo a transposition. So what I want you guys to do, the answers are posted on OWL, list all the mechanisms that generate genomic variation. And I think you've learned a lot of those mechanisms, if not all of those mechanisms in this class. Just list them down, the types of mutations, and how that leads to variation. All right, so I'll see you guys next week.